Okay. All right. Well, welcome everybody. I hope you all had a nice Halloween weekend. And getting used to the time change. I don't know about you all, but I absolutely hate the time change so much. It really bothers me. Takes me like 2 weeks. It's crazy. Okay, so I am going to go ahead and introduce our speaker for today. So Dr. Nation's research explores the intrinsic and extrinsic processes that generate and maintain species diversity and phenotypic disparity. He focused, you're challenging me with the vocab here. Okay, he focused primarily on small mammals in regions such as the montane tropical regions of Asia, the diverse subtropical landscapes of Florida, and the boreal forest of Alaska. But his research often spans broader geographical and phylogenetic scales. The Mammals Lab at the Florida Museum investigates morphological evolution and evolutionary ecology in various mammalian taxa, examining adaptations and diversity, accumulation in unique ecosystems. Their studies range from skeletal adaptations in climbing species to dietary macroevolution to phenotypic disparity within island communities. Their work focuses on small mammals, but their questions extend beyond any specific clade or taxonomic level. They aim to integrate foundational theories in evolutionary ecology with modern comparative methods, using a variety of statistical tools to gain deeper insights into evolutionary processes. Museum collections are central to the lab's research. They use a range of specimen data in their research, from external measurements and locality data to micro CT scans of fluid preserved specimens to DNA from frozen tissues to hair samples for stable isotopic analysis. The lab has an active collecting program in Florida and work in collaboration with several labs in Southeast Asia to inventory the astounding diversity in this beautiful yet threatened part of our planet. So thank you very much for being here.
Thank you. Apologize that I should have written that a little differently. Cool. All right, great. Thanks everyone for showing up. And everybody on Zoom as well. Cool. Yeah, this would be pretty, hopefully it's pretty fun. Great. Yeah, it's good to be here. So thank you all for coming. I'm just gonna kind of take this opportunity to really introduce myself. I'm talking to a lot of people who probably haven't heard me speak before. UF is massive, as you all know, and so there's all these different pockets of researchers that are actually doing overlapping research. So hopefully some of this is hitting, getting seen by some of the people who have yet to meet. So I'm a museum biologist. I've been working in collections for about 15 years. Started when I was an undergrad doing, preparations, entering data into computers, moving drawers around collections, all the sorts of things that one does when they're working in a museum. I did this as well as a graduate student, as a curatorial assistant, and then I worked as an assistant collections manager as well. After finishing my undergrad. And so all this time around mammals and mammal specimens and other things, one of the things that's always been really cool to me is how many surprises there are. A lot of people think that with mammals, we know so much about them compared to, beetles or other invertebrates or something. The fact is we actually are constantly learning new things about mammals, and they're always doing things that we kind of don't expect them to do. My undergrad advisor would always say, they don't read our books. They don't know what they're supposed to be doing. They just do their thing, right? And so we find animals sometimes strange places, sometimes doing strange things. So as a grad student, I started working in Southeast Asia, mostly in the Malay Archipelago, which you can see up in the upper right of the screen there in that map. And this really introduced me to biological diversity on a really different level than I had experienced before. It's an undergrad in Alaska. So I learned all about lots of cool different mammals, rodents, as well as shrews and other kinds of neat little things like on the left-hand side. Let me try this out here. Here's our pointer right here. That is a gymnor. It's basically a kind of a hedgehog, except it's soft and furry rather than spiky. And this work and some of my interests have led me down kind of a variety of paths that all kind of connect around questions of ecological disparity and phenotypic disparity. So I've been interested in the effects of environmental variation on phenotypic evolution. A lot of this has been framed inside of ecogeographic rules. Some of you may have heard of these. Bergmann's rule, at least in vertebrates, is a commonly referenced one. The idea that animals should be larger in colder and or wetter places. So my colleague Emily Roycroft and I tested this within some endemic radiation of rodents in Australia. Also working with all kinds of different tool sets and data types. It's ranged from like CT scan data, lots of external measurements to things like stable isotopes, which I used to understand a little bit about how dietary diversity is distributed among clades and communities in some rodents in Southeast Asia. And if you work in parts of the world like the Malay Archipelago, you inevitably run into problems with alpha taxonomy. You wind up catching something and you're like, oh, wait, what is this? And turns out it doesn't have a name yet. So I've sort of been pushed into a lot of molecular systematics and alpha taxonomy just due to that. It's not really the focus of my research, but it's really important because you have to know what you're actually looking at to be able to answer any questions. So that history and relationship among organisms is really critical whenever you're asking questions about, evolution on sort of any level. So I sort of think of my research as kind of meshing these three aspects together. Functions, so how an animal moves and how it looks, more of a phenotype perspective with history. And I have a tree there, so that means phylogenetic history, so relationships among animals. This also can mean like the history of the land masses that they're inhabiting, because the biogeographic history of these organisms is really important as well. And ecology, so how the animals interact with their ecosystems, other members of their community. And so all these things connect together to help us understand organismal form, phenotypic diversity, and put kind of simply, how animals evolve to do whatever it is that they do on a daily basis to get by. So kind of really broadly, I've had also to think about this question that's sort of been persistent, and it's going to be the focus of a lot of the talk today, about how we actually describe and define various aspects of an organism's biology and their behavior and their ecology. How can we describe the different things that these animals on the screen do in a way that actually, we can use in like to analyze some data, right? In a quantitative way. How do you take all the different ways of moving, types of food and everything and turn it into real data that help us understand processes better. So I'm going to talk about two pretty critical axes of ecological variation in mammals, at least today. I'm going to focus on locomotion and diet. I'm just going to briefly touch on locomotion for a little bit and then spend most of the time talking about diet in mammals. These two are related, of course. An animal has to move around. So locomotion means like the mode of which the animal moves around, right? So arboreal locomotion is moving around in the trees. Fossorial locomotion would be what this mole is doing in the bottom right, digging through the dirt and tunnels. And so the animals have to move around to get to the food that they need. So these are connected, but they each have kind of their own challenges as well when you're trying to understand them better. So starting first with locomotion, I got interested in locomotion as an undergraduate, actually, looking at this species called the northern redback vole, Myotis rutalis. These are really abundant and widespread taxa or species in Alaska, as well as kind of the circumpolar north. They're all over the place, but we had gotten some anecdotal reports of these animals climbing up in trees. And this was something that they weren't really known for doing or supposed to be doing. We searched the literature and found no instance whatsoever of this animal climbing in any literature. And so this was weird. And so my undergrad advisor tasked me. He said, okay, go figure this out. So I did a lot of trapping and also camera trap work. And it turns out that these animals do climb and they're quite good at it. We caught an animal on a video outside of Fairbanks in the middle of winter. It was about 20 below that day. This vole popping out of the snow, climbing up a spruce tree, eating lichen that was up in the tree, and then climbing back down into the hole. And we don't have any information on the diet of these animals in the wintertime, but we know that in the fall, they start adding more lichen into their diet. So we suspect that actually in the winter, lichen is one of the few potential foods even on the landscape in interior Alaska in the winter. And perhaps they're actually climbing to access this particular food. The thing about it, though, is that there's nothing about this vole. They're about yay big. For those of you who don't know much about small mammals, there's nothing about this animal that makes you suspect that it's good at climbing. It has short, stubby legs, which is the opposite of what we'd expect for a climbing animal. Has a short little tail. Um, but, regardless of how it looks, it actually is quite good. And so moving on to murine rodents, which has been the focus of my work for about the past decade. This is a subfamily of rodents that contains about 700 species. So that's right now about 12% of all living mammals are in this one subfamily. So there's a lot of them, and that's definitely an undercount. There's certainly a lot more species that are yet to be described around the world. Most of the diversity is in sub-Saharan Africa and Southeast Asia. And they do all kinds of things. They swim, some of them climb, some of them dig, they eat all kinds of foods. And interestingly, deeply nested within this one radiation of rodents are both Rattus rattus, so the lab mouse, and Mus musculus, the lab rat, I'm sorry, and the lab mouse. So these two are part of that radiation, but the radiation itself is much broader and really ecologically diverse. And locomotion, I think, plays a major role in the success of this group and how communities of different murine rodents are formed. In one study I did with a bunch of collaborators, we looked at different montane communities of murine rodents on the island of Sulawesi in Indonesia. It's there highlighted in the map in the bottom. And each of these mountains, shown as the little circles on the map, have a community, their own community of murine rodents. And the number of species ranges from about 7 in the least species rich up to at least 24 species. 24 closely related rat species, all living together in the same place on this mountain. And so that really, you start to question the idea of competitive exclusion, right? How are these things all interacting with each other? How are they all able to live there and coexist? And we tested a bunch of different traits to test the ideas of niche expansion and niche packing, like how are they able to fit so many species into a community? And turns out locomotion seemed to be the most important way. There's different rodents there that actually will eat very similar foods, but they do it in different places. So you can eat invertebrates in the water, under the ground, on the surface of the ground, or up in trees in these kind of montane habitats, right? So locomotion and the diversity of locomotion within locomotor modes within a community turned out to be really critically important, more so than other types of ecological traits that we measured. But locomotion is really hard to define in small mammals. I mentioned that we have things like categories like arboreality or terrestriality that we use when we're describing them, semi-aquatic. But these rodents don't always fit into these nice little boxes, these nice little categories. I'll give you an example. This is Linomys meyeri, the nice, I think, a quite handsome rat found in Sulawesi. Long woolly fur, has a lot of traits that you associate it with arboreality, with climbing. It has kind of long fingers, but they're also quite fat and have big pads on them, so it increases the surface area of touching the branches. Long tail, short, very curved claws that can grip into the branches. And also, it does climb. We know it climbs because they are frequently caught up in trees. However, one researcher who spent a long time in Sulawesi back in the 70s caught one of these and let it go and tracked it to see where it went. Turns out this animal, this arboreal animal, went into a burrow. The man who doing this guy must have excavated this burrow. Turns out this climbing mammal was in a burrow longer than 5 meters long. It had a nesting chamber in it, various like false entrances. We don't know if this animal dug this burrow, but we know that this animal that we would obviously think is arboreal also lives in very intricate burrow systems. So all of a sudden, this idea of calling this animal arboreal kind of falls apart, right? Is it arboreal really? But it lives, a bunch of its life down in a burrow. We don't really have a way of quantifying that. How do we talk about that? We don't have a category of underground and up in a tree, And so just with that in mind, I'm going to move over to diet, and we're going to kind of keep that keep that focus going of how we talk about different characteristics of ecology. So this is, in a large part, a study um, working with Graham Slater and Anna Wisniewski, who are Graham's at University of Chicago and Anna's now a postdoc at the Museum of Paleontology at Michigan. So diet's really a fundamental aspect of species ecology. We know that communities are often assembled on axes of dietary variation. And dietary preferences can lead to phenotypic change, right? When an animal eats a certain food, they need a body that can digest that particular food. They need to be able to access that particular food, right? And so this is really important in the overall accumulation of diversity on the planet. It's very important, but how do we measure diet? Well, similar to locomotion, we often use categories like herbivore, carnivore, omnivore. And you don't have to look far below the surface to realize that these are kind of insufficient, right? We have a hyena here and a mole, and they're both carnivores. There's nothing at all overlapping ecologically about a hyena and a mole, right? But they both fall into this category. So one way you can handle that, people often do, having a little bit of font trouble now, that's all right, is create lots more categories, like finally partition these categories, right? We have a frugivore granivore, like this chipmunk, a frugivore omnivore, like this monkey, a large mammal hypercarnivore, like a hyena. And this can work sometimes, but the problem is when you start including lots of different species, you end up with a ton of these categories that are quite narrow, and there's not good representation of each taxon of species in each of these categories, right? You may have nectarivore as a decent category, and it might work for a small subset of bats, but if you try to expand that out to mammals, you don't really have enough nectarivores to really be a meaningful category in all of mammalia. The other thing is that these all focus on primary foods, right? Like the main food that this animal is seen eating. But animals eat all kinds of foods. And they have these secondary foods and other things that are important seasonally. I like to show this video, if it plays. So this is a ground squirrel eating a lizard. Chowing down. This is taken somewhere in Western North America. And so squirrels eat meat regularly, somewhat regularly. It's kind of weird, I know, it's kind of gross maybe, but they do. They're known to eat meat, but they don't eat it all the time, right? And you would never say that a squirrel is carnivorous. There aren't any real carnivorous squirrel species out there. But many species are known to eat either vertebrate or invertebrates, at least occasionally. Like there's something really important about this food for them, either maybe seasonally or we don't really know, but there's something critical in that. But how do you include this in when describing a diet of a squirrel? We know they eat mostly seeds, but there's no way to have a category that focuses on mostly seeds, but also lizards, Um, so I like to share this quote from John Eisenberg. Classifying living mammals into categories according to dietary preferences is fraught with difficulty. Mammals, like all other living organisms, have a perverse tendency to defy exact classification. And he's right. This isn't a new problem that I'm talking about. This has been something that people have been thinking about for a long time. John Eisenberg was the first Ordway Chair of Ecosystem Conservation at the Florida Museum. And this is, he started here in '82, a year after the mammalian radiations came out, which is a really excellent book that holds up today. In this book, he actually went through and came up with some ideas of different categories that we could use. He thought really hard about this, but he prefaces that chapter recognizing that it's almost a fool's errand to even try to do this because things are just so hard to classify. And I know that no matter what all of you do, most likely you deal with classification at some point where you're forced to lump something into a group and it's never like a perfect fit, Everyone I've talked to in any sort of discipline has to do this at some point. And there's many complex things that our human brains can't really handle. And so we classify them, whether it's like types of cars or like types of diet or whatever it may be. So it's really, it's a pervasive problem. So challenges with diet, well, again, information loss and the resolution in categories. the low quantity foods are difficult to incorporate. With diet in mammals, we get all kinds of data types. Like a lot of it, we get observational data, diet data would even quite look like. If we could even somehow magically record every single thing that an animal eats, get the caloric content, the protein analysis of all these foods, it would be so incredibly complex that I wouldn't even know where to start with how to analyze it. So this is an idea that we borrowed from some other people who sort of introduced it, but didn't really take it very far. An idea that Graham and Anna and I had of another way of talking about diet. So rather than trying to categorize the animals themselves, we can try to split up diet into a bunch of its own categories. So having food items, we call them, but these are essentially food categories, right? So here on the left are four mammals, and we want to describe their diet. There's a lion, a hyena, a raccoon, and a coyote. And rather than categorizing these animals, what we can do is have these diet categories. So in the first column here, that deer represents a food large mammal. Then we have rabbit representing small mammal as a food type, bird as a food type, hard invertebrate as a food type, and then fruit as a food type. Then what we can do is actually rank how important each one of those foods is to that particular species using this ranking system, right? So 1 means that this is not an important food or it's just not known to eat this at all. 4 means that this is a critical part of its diet, right? These are real data, just a subset of the bigger data set, but so raccoons and coyotes have both been called omnivores before. You see that there is some overlap. They both eat a lot of small mammals, but there's also a lot of difference in their diet as well. Raccoons rely a lot on hard invertebrates and fruits, and coyotes don't. So this is actually pretty information rich comparatively, and you don't need a lot more data to do this than you would to actually categorize a species. It's pretty flexible for taxonomic groups, so you can sort of have as large or small as a phylogenetic scale as you want. You focus on all of Mammalia or just a subset of one rodent clade if you wanted to, and you can just adjust your categories accordingly. And it's easy to include these low quantity foods. When something eats, like for that squirrel, for lizards or small mammals or whatever, we might give it a 2 because we know it eats it occasionally, but it's not a major component of its diet. It's easy to deal with interspecific variation in this way. And ordinal rankings. So these are ordinal rankings, essentially. And there's a lot of data analysis tools to deal with that. Ordinal rankings are really common in social sciences, marketing, other things. These are essentially Likert scores, right? So how likely are you to recommend this seminar to your friend? Very likely, somewhat likely, not likely at all. Those are ordinal data, ranks, right? And they can't be treated like linear data. It's not like a 1, 2, 3, 4 on a linear scale. They're different, but there's tools to deal with that. So we constructed a matrix of 13 different food items for 90 different carnivorans. Carnivorans, that's the mammalian order Carnivora, so like cats and dogs and bears, weasels, other things like this. And we have 13 different food items that we use for this study. That's a list of them there. And this was really, again, focused on food items that we would use for carnivorans, right? If we were including other things like, primates or rodents, we would probably split these up and partition these a little bit differently. And this is actually the data set there in the middle. That's the whole matrix of ranks right there. One thing we can do is actually take these ranks and project them into what we call a diet space, like a multivariate or multidimensional diet space. You can use a tool called polychoric PCA, which is just the kind of principal components analysis that is geared towards these kind of ranks rather than continuous data. With this diet space, then we can start to ask questions about diet that we haven't been able to before. Like, what are the primary axes of dietary variation? How does diet disparity differ among clades? where are dense and sparse regions of diet space? What are diets that have evolved a lot or diets that are somewhat rare in different groups? And how is evolutionary history reflected in that? These are the kind of questions that we can ask with, phenotypic data or maybe ecological data like climatic data, but we haven't been able to do it very well with diet data because we've just been sort of stuck in these categories. So this is what the first two axes of a diet space looks like. I put the four kind of our four focal animals where they fall out there in diet space. I also put this phylogenetic tree up there. This is just kind of showing two of the main clades suborders within carnivorans, the feliforms and the caniforms. The feliforms are cat-like animals, cats and hyenas and civets and things. Caniforms are dogs and weasels and bears, raccoons and stuff. So the first axis, which is 28% of the variance, really splits off these large mammal consumers versus like the invertebrate consumers and the everything consumers over on the left. So that's really the biggest axis of variation in the diet among these carnivorans. Whereas axis 2, 18% of the variance, pulls out small mammal and bird consumers on positive values, and then fruit and plant and other consumers down here in the bottom. So this is about 50% of the variation in the data set, which is quite cool that we can really just represent on these two axes. But remember, there's 13 categories, so there's 13 axes to this multidimensional data set. So one way we can look at how diets are distributed then is do something like look for clusters in the data. So we did a k-means cluster analysis. Turns out 2 clusters best represents the data. I colored those in yellow and green here. You can see them. And then I plotted those up on the phylogenetic tree. And so we can see that both of these clusters are pretty equally distributed among the 2 different major suborders of carnivorans. This tells us that these different diets or these dietary clusters have evolved multiple times within carnivorans. And then I also showing these outlines up here. Up here are hypervolumes. So these are multidimensional volumes. One's for caniforms, one for feliforms. And you see that they both are quite large and they overlap a lot. So this is telling us that each suborder actually occupies a pretty similar breadth of dietary space. And this is actually contra what a lot of research has stated in the past about differences in dietary evolution between like cats and caniforms. But because we're able to look at it in this interesting multivariate way, we're kind of gaining a new perspective. Another thing we can do is look at the density of diet space. So how densely packed and sparse are the different regions of this multidimensional space. And so that's what we did here. So these blue cells that are wrapped around the tree, don't spend too much time trying to focus on them or figure out exactly what they mean. Basically, dark, that means it's a really common diet, and light means it's a really unusual diet, basically. Then we can take this density and also determine whether or not the other animals that are really close to it in diet space are closely related or are they distantly related. And this can tell us more about the evolution of certain diets. Like, is this particular diet useful in the success of one group, or is it one organism that evolved a diet that's really dissimilar from all the others, all its other close relatives? I'll kind of walk through a couple examples of different possibilities here of what that could look like. So A here, I highlighted on the tree and also in the multivariate diet space up there. This is a kit fox. This is a cool little fox from North Africa. It's in a really dense region of diet space. So there's a lot of other carnivorans that have a diet very similar to a kit fox. But most of the animals that have a diet similar to a kit fox are other foxes. So that's what I mean by when I said phylogenetically clustered diet. It comes out as being very clustered. So a bunch of other foxes are also found in that one area of diet space. So foxes are shared this very similar diet, and there's a lot of them. Opposite that would be a binturong. This cool, like arboreal civets in Southeast Asia eat a lot of fruit. So they're in a really sparse region of diet space. So there's not many carnivorans that eat the kinds of foods that binturongs eat. And it's a phylogenetically over-dispersed diet. This means that the animals that do eat foods like a binturong are not close relatives. This binturong is really off on its own. All of its close relatives are doing something totally different. It's just figured out how to have this diet of eating fruits and other things up in trees. The other ones that are similar are actually like kinkajous and some other raccoon relatives that are up in trees and eating a lot of fruits. This is a hog badger, a cool badger with this pig-like nose from Southeast Asia. So they're in a really dense region of diet space. So they eat invertebrates, like soft and hard invertebrates, which a lot of carnivorans do. So this is a really dense region, but it's phylogenetically overdispersed. So lots of different carnivorans from all over the tree have evolved this really invertebrate-heavy diet. And lastly is river otters, which are in a sparse dietary region. There's not a lot of just fish-eating carnivorans. I should preface that by saying we didn't include things like seals and sea lions in this study, just terrestrial animals. So there's not a lot of other terrestrial carnivorans that eat a lot of fish, but all the other ones that do eat fish are otters, right? So it's phylogenetically clustered. So this is something that just really evolved once. There's not a ton of them in this area. It's really sort of an otter specialty that they kind of did on their own. So another thing we can do is try to connect diet to phenotype, which is what a lot of us probably want to do who study, plants or animals or whatever, is try to link something about an ecology to something about the structure of the organism, right? Whether it's like leaf shape and how that fits into the environment, or in this case, tooth shape and how that fits in with the food. So we use these carnassials as a model system. The carnassials in carnivorans, a lot of C words, it's kind of this model system. And it's been cited as a key innovation sometimes for carnivorans to open up this new door of eating a new kind of food, like eating big mammals, like eating gazelles and moose and other things, because they have this really powerful toolkit in their mouths. This carnassial, this is called the carnassials here. It's the upper premolar and the lower molar. And in a lot of carnivorans, it makes basically a pair of scissors, like really, really powerful scissors that can slice through, outer skin and slice through meat, allowing them to actually process, things like a moose or a water buffalo or something. But not all carnivorans have this exact shape. A lot of them have teeth much more like ours, like bears and raccoons. Their molars kind of look like ours. They're really good at grinding up things, right? Not slicing things like scissors. So we can take this shape variation and use regression models that are meant to be used with ordinal data to better understand the relationship between particular foods and the shape of teeth. I'll add in that we also, body size is really important. Like the size of the organism is really important in all these kind of studies. So we actually use body size as an interaction term within this. I'll explain a little bit more about what that means in a minute if you're not familiar with interactions in linear models or regression models. So the example I'm going to show, just to preface it, the food I'm going to be talking about at first is large mammals. That's like the category of food we're talking about. Think deer or again like moose or water buffalo or ibex or whatever. The phenotype is the grinding area. So this is basically how much of the tooth is used for slicing versus how much of the tooth is used for grinding. And this varies a lot on carnivorans. Like cats, it's all for slicing, whereas like raccoons, it's mostly for grinding. So this is what the results of these kind of models look like. On the x-axis here, we have our phenotype. So this is the grinding area measurement. So the ones on the left, for the left values, those are really sharp teeth, and the right values, those are really dull teeth. And then on the y-axis, we have probability. So this is the probability of belonging to a particular rank. Okay, so the probability of being in that rank. So we see animals that have this large grinding region over here have a very high probability of being in rank 1, which means they probably don't consume large mammals. Whereas on the left-hand side of that, you see the really sharp teeth, the probability of eating some or maybe a lot of large mammal increases a lot, right? Which kind of makes sense. But I mentioned that we use mass in these as an interaction term. So what that means is that this relationship between phenotype and diet can actually change depending on mass. The model allows for that to happen. We actually find that happens quite a bit. So on the far left side, this is for small-bodied carnivorans. So we set this to be 1.5 standard deviations below the average in our data set. And we see for the smaller animals, it doesn't matter how big what your teeth look like. You just don't eat a lot of large mammals, which makes sense, right? Smaller carnivorans aren't able to take down things like water buffalo. But if you're big, over here with these high mass relationships, it really matters a lot. The shape of your teeth is really critically important for this particular type of food. So if you have really sharp teeth, you have a high probability of eating almost entirely large mammals. Whereas if you have dull teeth, a high probability of eating very little large mammal. So this relationship is actually changing depending on the size of the organisms, which I think is pretty cool. It's actually something that's overlooked a lot in a lot of these studies. Size is something, there's a whole discipline of evolutionary biology focused on size. But size is also often something treated as something that needs to be like controlled for. I kind of rarely see it used as an interaction like this, recognizing that the relationship may actually vary depending on the size of the animal. But we found that the tooth morphology is really good at predicting a lot of these different foods, but not all of them. Some of the foods we had chosen, teeth just don't seem to do a good job predicting, like eggs, for example. Don't seem to be any real phenotypic adaptations in carnivorans for eating eggs. A lot of them eat eggs opportunistically, and I think it doesn't take a lot to open one. But we also can, and we did this in the paper some, you can actually use these relationships and these models to actually predict the importance of different foods for either really poorly studied species or extinct species. And there, the paper is down there if you wanted to, investigate that a little more. That was some pretty fun stuff. So anyway, I think this method is pretty flexible, and we think it could be used with lots of other things beyond diet as well. So there's one study that hopefully will be submitted soon that we use this for environment or for habitat in artiodactyls. So like savanna, forest, grassland, wetland, and a couple others. A lot of different artiodactyls. This is things like, again, like deer and water buffalo and zebras and other things. I'm sorry, zebras are perissodactyls, but yeah, they have a habitat that they often use a lot, but then they will bounce around and use other habitats preferentially or seasonally. And so this actually worked really well for looking at habitat as well. We're trying to apply this to locomotion. There's some tricky things about locomotion, like whether something climbs to eat, or if it climbs to nest, or if it climbs to run away. And all those might result in different sort of phenotypic adaptations. But we're working towards getting some sort of framework to actually look at locomotion in a more multivariate way as well. So yeah, I think that this has promise. I do want to say that we don't think that this is the only thing to do or the way that everybody has to do everything from now on. I think that many people, no matter what you study, are probably occasionally just a little maybe distrustful or uncertain about the categories that you have to use in your own research. And so the idea is really just to kind of get a conversation started about how we can get around that and actually use complex multidimensional traits in our data analysis and things. Cool. So I spent the last few minutes talking about some of the work going on in the Mammals Lab at the Florida Museum. So one thing I didn't talk about today, but it's been a big focus of my work lately, is looking at the ecomorphology and evolution of shrews in Southeast Asia. So shrews are really interesting for a lot of reasons, but one of them, in this part of the world, they're really, really diverse. There's a lot of endemism, so many of these species are only found on a single mountain. And there's also, in some places, a lot of shrews living in the same place. Like one mountain in Sulawesi has actually 10 shrews on it. What's crazy about shrews, even in comparison to rodents, is they all look a lot alike, right? It's really hard to tell just by looking at them what they could possibly be doing differently from one another ecologically. And the fact is, we don't know. They're hard to observe. We don't have any observational data. We don't know how they're able to coexist in a single place without, again, back to the rules of competitive exclusion. You're not supposed to have, more than one, like two closely related species that are doing the exact same thing in the exact same place, right? But that kind of seems to be the case with these shrews. So we're digging a little deeper, trying to look at things like skeletal morphology and other ecological components that we can get from museum specimens to try to understand this a little better and see if they might be, maybe moving through their habitat differently or something like this. Ellie Bollock, a student in biology who just started this year, is going to be sort of carrying the torch for a lot of this kind of work and also working on a lot of the systematics. There's several species that need to be described as well, and she's working on some of that now. I'm missing some dots here on the map, but that's all right. That's Alachua County and Marion County. We're also kind of, as I speak, getting some trapping off the ground here in Florida. That map in the the plot in the lower left shows in the black line is the number of specimens from these two counties in the collection at the Florida Museum. And then that's the population growth chart of Alachua and Marion counties. And so it's changed a lot since the 1960s when both of these had under 50,000 people in them. But we have a lot of specimens in our collection that were collected in the late 50s, also in the 70s. And to study change, you need at least 2 points, right? You need a before and you need an after. And that's really where museums shine. And that's what we do. We have historical data that can be used in comparison to modern data to better understand how things change over time. One of those projects we're working on is looking at shrews in Florida as well. There's this really funny species in South Florida called Blarina shermani that's only known from a single location. It was caught in the 50s. It's now a species of concern for the state. No one seems to be able to catch it at all anymore. We don't know if it's actually a valid species. So preliminary work suggests that it's not actually anything different than the other shrews that live in Peninsular Florida. That's really been spearheaded by Verity Mathis, the collections manager. But there's some other interesting things as well, like whether or not the ones in the rest of Florida are actually different than the ones that are in the Southeast at large. And so whether the species peninsula is actually legitimately a thing, seems like they are, likely hybridize a lot if they are actually different species, but we know there's different chromosome numbers among some of these populations. So a bunch of mysteries there that will be really fun to work on. And Ellie's also helping out with some of that. So Meg, who started here in SNRE this fall, is really interested in color in small mammals. And so she's starting to work on background matching and crypsis in 6 different rodent species in Florida, looking at the change among different types of habitats. So these are actual museum photographs. The screen's pretty bright here, but you can probably still see the differences in color between these Paramiscus taken from a scrub habitat versus more of a hardwood hammock habitat. No one's tried to quantify this variation and seen if this variation actually exists among lots of different species. There's also some hypotheses about how much heterogeneity should exist in a habitat. Some of these like fire-driven ecosystems, they can change color really quickly, right? A fire blows through, all of a sudden it goes from green to black, and then it slowly goes back to green again. So the idea is that it might be beneficial for a population to maintain more color heterogeneity in a habitat that changes frequently. And we don't know if that's true or not, but it's an interesting thing to test in in some small mammals, and so Meg's going to be working on that. Brooks is an undergraduate student who's interested in climbing ability in some Florida mammals as well, some Paramiscus, Potamies, and Ochratomes species. Trying to figure out if there's any differences between different habitats, differences just between the species, and maybe get a grasp on how plastic some of these phenotypes are, some of these things that relate to climbing. There's some evidence in like lab mice that, just in one generation, depending on the substrate they're moving in, there's actual quantifiable changes in the skeleton. And so we don't know how that actually plays out in the wild. And lastly, Chloe, who's another undergraduate in zoology, is working on quantifying fluctuating asymmetry in the skulls of Florida panthers. So we have a really phenomenal Florida panther collection at the museum dating back almost 100 years. Developmental instability, so inbreeding and stress can lead to problems in development, right? And this often presents as something called fluctuating asymmetry, which is where the skull or some body part ends up being asymmetric rather than symmetric, right? We're supposed to be bilaterally symmetric, at least our skeletons are. There's always a little variation, but that variation massively increases under times of developmental stress. And so quantifying that over the past century to see how that has changed and perhaps gotten better since the genetic rescue took place with that population. Yeah, and I guess lastly, our job at the museum is to support people in their research. We have the project we're working on, but we also have a collection of about 40,000 mammals. If any of you have any interest in anything that might could use our collection, let us know. No project is too small for us to, talk about or take on. All kinds of possibilities. We have a lot of historic specimens. And so again, we have time points in the past, little time machines that you can use to look at what an animal looked like 50 years ago or 100 years ago as compared to now. Um, Studies with DNA or maybe with stable isotopes or just distribution modeling of different species. And really limits only our imagination with what we do with these museum specimens. So yeah, please contact us if you want to know more about the collections and how that can be used in your research or anyone else's. That's why we're here and what we like to do. So just a thanks to SNRE for setting this up, and then some funding sources and other collaborators. And yeah, and if there's time, I can take some questions.
I've got a question.
All right.
So the Florida Panthers
Mm.
thing that your student is working on, do you think that she's going to find? So I just this is me as a non-biologist asking this, but I know that they brought in the Texas cougar, right, to
Yeah.
sort of diversify the gene
Mhm.
pool there. And so, do you think that she's going to sort of be able to notice that if she's looking at sort of this longitudinal set of data?
Yeah, I don't know. That's kind of the main thing are interested in, right? And so, I'm new to this. I don't have a history of Puma research or anything, but there were a lot of phenotypic problems with the Panthers when they went through that massive bottleneck, right? And the ones that are commonly discussed are like the kink tails and the cowlicks. A lot of those have gotten better from what I've been told. We had all that material used to come to our museum as a state repository. That connection was a little broken in recent years, but we're trying to build it back up so we can at least continue taking in panther specimens as they, most of them are hit by cars and other things, right? But it's better that gets saved rather than, incinerated. Um, and so, yeah, that's my basic hypothesis is that would be the case. But I don't know. Yeah, looking at these skeletons, though, a lot of them are, they're weird. A lot of these really the ones from like the 90s that were highly inbred. You just kind of look, and if you know what to look for, it's like this is something strange here. And so yeah, that's my guess. But yeah, I'm not quite sure. Yeah, it should be interesting to find out. Yeah. Okay.
Super interesting talk. Thanks. Two questions. Do you, so for the shrews, I know that some small mammals make really interesting high-pitched noises that we can't hear. I don't know if this is something you could study from a museum standpoint,
Yeah.
but they might have differences in their ability to hear things. That
Yeah. No, that's actually really interesting. Yeah. Yeah, that's cool. I like that, and I think I might actually continue on that. So there's a whether the hyoids touch like the tympanic bone in the skull is an indicator of echolocation a lot. Bats have this connection between their throats and their ears, essentially, like a bone connection. And that doesn't exist in non-echolocating mammals. And in shrews, in some shrews, it seems to be there. I don't know if that's been quantified in any of these like Southeast Asian ones. But it's very true. I actually have another colleague who works on larynx musculature in bats, more of a physiology side. But he's going to be down here soon. I'm going to talk to him about differences in like larynx structure, other things as well. Yeah, that's cool.
Yeah. So the other question I had is sort of technical about the PCA plots that you're making. Did you have a method of placing the showed the picture of the different carnivores over their
Yeah.
Did you?
That wasn't precise. I just kind of put them in there where they, let's see if there's one. Yeah, no, I just kind of put them in there more or less where they go. So I just sort of added those little silhouette images on top of there. I think the hyena is actually up a little bit, but I just sort of put it there for readability.
Okay.
They're kind of like, yeah.
So how about when you draw the circle around the area where you think it's carnivore versus the
versus the two
different
clusters. Yeah, so those are just really clusters in the data set. Let's see, did I have one of those in this section here? Hold on one sec. Oh, hey, here we go. Cool. Yeah, so those are just clusters in the data set. So for cluster analysis, you're basically just asking like, is there any pattern here? It's like an unsupervised method of seeing if there's any natural clusters within the data set. So it's just the way, yeah, go ahead.
Does it how would you decide where to cut it off? Are you just approximating it, or do you?
With the plotting?
Yeah.
Yeah, so I think I used I think so in R, like so this is done in ggplot, and so I think there's several methods. So you could do a convex hull, or you could do an ellipse. I think it's an ellipse, and I set the ellipse at I think it's like 90% or 95%. Yeah, and so that's what forms that. Yeah, that's what forms that circle. Right. I find that a little easier to read personally than doing like a convex hull and having kind of the jagged edges. But there's different settings within that whole plotting feature that can get sort of a smoother, rougher surfaces and stuff like that. Yeah.
It's kind of based on a density.
Yeah, exactly. Precise. Yeah, and you can also do one that looks more like a topographic map as well, where like, it's zooming in on the peaks and stuff too. So, yeah.
Okay, I think that's our time for today.
Great.
And maybe if anybody has some lingering questions, if you have a couple
Yeah.
minutes to hang out. Okay, cool. Well, thank you to everybody on Zoom for being here, and thanks to all of you. And I will see you next week. And for the people in person, there's some persimmons down here that are oddly shaped. If anybody wants one, please help yourself. All right.
Cool. Thank you, guys. Yeah, stop share.
Sure.
Stop share. Cool.